So this is part 2 of our lecture, finishing up chapter 48 and also covering chapter 49.2 from your textbook. So we left off talking about what happens as a neurotransmitter crosses a synaptic gap and binds to the postsynaptic membrane, and in that way causing some sort of either excitatory or inhibitory postsynaptic potential. So there is some sort of response that's going to occur in the next cell that receives that message via the neurotransmitters. But what happens with the neurotransmitters that are released into that synaptic cleft? There have to be mechanisms to clear them out to limit the duration of that postsynaptic potential. So some neurotransmitters are simply going to diffuse out of that synaptic gap. Some are going to be taken up by surrounding cells. Some are going to be, and that's going to be through active transport and repackaged into synaptic vesicles. Glial cells also actively take up neurotransmitters at some synapses and metabolize them as a source of fuel. And then finally, there are enzymes that are going to catalyze the hydrolysis of the neurotransmitters. They're going to break them down. In particular, one of those neurotransmitter enzymes, this is actually the wrong slide. This is the one I was looking for. One of those enzymes that breaks down the neurotransmitters is acetylcholinesterase, and as you might guess, the neurotransmitter it breaks down is acetylcholine. And so what we have, an example of how some neurotoxins actually function is the example of the snake toxin that actually, snake venom, and also sarin, which is a neurotoxin, and some insecticides all work by inhibiting acetylcholinesterase. So what they do is they prevent the removal of the neurotransmitters and cause a sort of paralysis from the hyperstimulation that would occur when you have this continual message going across the synaptic cleft. Going back to that last slide, what that was talking about was how neurotransmitters are a weak point of the nervous system. Because of what we just were describing, there are many substances that can mimic them and in that way inhibit or cause problems, hyperstimulate the nerve in terms of its function. So some of those things that cause problems are gases like nitrous oxide, used by dentists to relax people who have fear of having dental work done. The way that works is by affecting the neurotransmitters. Carbon monoxide, as you I'm sure are familiar with, that causes it's lethal, it's a fatal gas that will block a neurotransmitter's function. We already talked a little bit about how drugs can do that, both stimulants and depressants. Caffeine actually affects the function of neurotransmitters. Nicotine from cigarettes, amphetamines, quaaludes, barbiturates, all of these are going to have their action by mimicking a neurotransmitter. And then we have the hallucinogenic drugs like LSD or peyote, MDMA. Some of these prescription actually mood altering or remedying drugs like Prozac, Zoloft, Paxil. Those also mimic neurotransmitters. And then we have the poisons that we already talked about, like the snake venom. So that's it for chapter 48. Now what we're going to move on to is chapter 49, section 2. And that's going to be looking at the brain. So this is an X-ray showing the structure of the brain in the human adult. And essentially, the human brain contains about 100 billion neurons. So we've been talking about how neurons function on an individual basis or from one neuron to another. But when we're looking at just the central nervous system and actually just the brain of the central nervous system, we're talking about 100 billion of those neurons functioning and doing their job of having that action potential travel down their axons onto the next neuron. So these have to be organized into circuits um, or into some sort of networks that allow them to function in an efficient way. And nowadays and over the past several years, different imaging techniques are allowing researchers to monitor different areas of the brain and see as a person is actually doing something, see in what part of the brain is actually working together. So, how are those neurons coordinated to actually allow us to do the things that we do with our brain? So, section 49.2 is going to look at how the vertebrate brain is regionally specialized. So, we do have sections of our brain that are specialized for the different functions. But all vertebrate brains develop from three embryonic regions. We have a forebrain, a midbrain, and a hindbrain section. And by the 5th week of human embryonic development, 5 brain regions have formed from these first 3 embryonic regions. So we have the forebrain, allowing the formation of the olfactory bulb and the cerebrum. We have the midbrain, and we have the hindbrain here with the cerebellum. This is a good diagram showing how development of the brain occurs from an embryo at 1 month to the adult brain. So if you think about the specialization that's occurring here from these 3 portions, fore, mid, and hind brains, into this complex structure we have in adulthood, it's really pretty incredible. And these are some of the terms. You don't need to memorize all of these terms of the different portions of the brain. We'll look at some of the key functions and areas of the brain that you should be familiar with. So the forebrain basically has activities such as olfactory, which is our smelling, input, the regulation of sleep, and of learning, which, so your forebrains are busy this time of year, all year, and also complex processing. Whereas the midbrain coordinates sensory input and how that's going to be handled. And then the hindbrain is going to control involuntary activities and motor activities. So our coordination and also our sympathetic parasympathetic system that is involved in involuntary activities. Comparison of different vertebrates shows that the relative sizes of the particular regions that we're talking about actually is going to vary by the species and the different types of vertebrate, and it's going to have to do with how much function is needed. So for example, how important is sensory input to the actual organism as opposed to its motor activities? So that's going to determine the structure. And so again, our familiar phrase that our structure and function are very closely related. This is showing evolutionary relationships here. So we have an ancestral vertebrate, and then we have the different branching off of the different organisms here, from mammals down to the lamprey eel. And you can see that there's still this 3 forebrain, midbrain, hindbrain commonality, but the relative sizes of these different structures varies dramatically. So during embryonic development, the anterior or front neural tube gives rise to the forebrain, midbrain, and the hindbrain. And the midbrain and part of the hindbrain are what are going to form the brain stem, which is going to join the brain with the spinal cord at the base. The rest of the hindbrain is going to give rise to the cerebellum, and then the forebrain is going to divide into the diencephalon, which forms endocrine tissues in the brain, and the telencephalon, which becomes the cerebrum. So this is showing that developmental division of the different sections of the brain here. And again, you can find these diagrams in your textbook and studying them to become familiar with just kind of a basic familiarity with the structure of the brain. As the human brain develops further, the most profound change is going to occur in the forebrain, which gives rise to the cerebrum. The outer portion of the cerebrum is called the cerebral cortex, and it surrounds most of the human brain. The brainstem, you can see here, coordinates and conducts information between the brain centers, and it has three parts. The brainstem has the midbrain, the pons, and the medulla oblongata. So the midbrain, those three different portions of that brainstem are divided again by function. The midbrain is going to have the center for the integration and the receipt of sensory information, which makes sense as it's coming up through the spinal cord from the peripheral nervous system into the central nervous system. We have to have that sensory information processed. And so that's going to be received and processed in the midbrain. And then the pons regulates the involuntary things like breathing. And the medulla oblongata contains centers that control things like breathing, cardiovascular activity, swallowing, vomiting, digestion. So again, things that we don't have to think about and process are done and handled in this entry portion of the brain. The diencephalon develops into three regions, and these are things that you have heard of, like the hypothalamus, the thalamus, and the epithalamus. So the epithalamus has the pineal gland, and it generates the cerebral spinal fluid from the blood. And the thalamus is the main input center for sensory information and kind of the gateway for that information to the cerebrum and how it's going to be processed. It's also the main output center for motor information leaving the cerebrum. So it's kind of like the way station for sensory input, and then the cerebrum figures out what to do with that sensory information and then tells the thalamus to send out a message to the peripheral nervous system to do something if needed. And then the hypothalamus is involved primarily in regulating homeostasis and the basic survival behaviors like feeding, fighting, fleeing, and reproducing. This is a great diagram showing the locations of these different portions of the brain. So you can see here the central position of the thalamus, and that little blue dot is the pineal gland. This is the olfactory bulb. This is the amygdala here, and the hippocampus. And then this little portion right there is that hypothalamus. So one thing about the hypothalamus that's a very important function is how it controls the biological clock in our bodies. So the biological clock is a molecular mechanism that directs periodic gene expression and cellular activity. It is involved in what we would call our circadian rhythms, which are the daily cycles of biological activity, things like our cycles of sleep and wakefulness. And in order to do this, they need a center, an actual mechanism that's going to be able to do that. And so in mammals, there are a pair of what are called suprachiasmatic nuclei in the hypothalamus that actually function as that clock. And they do this by relying on external